Our next speaker is Derek Reiman.
Well, thank you for the introduction. My name is Derek Reiman. I am from the University of Illinois at Chicago, here in Chicago, so I don't have to travel too far. And today I'm going to be talking about a tool that we've been working on in my lab called PopPhy-CNN. And it's a convolutional neural network framework using kind of tree representations for analyzing the association of host and microbiome and phenotype. So hopefully I don't have to convince too many of you that the microbiome is an important topic of study, because it plays a fundamental role in host health. Just a couple examples: it's been shown to stimulate the development of secondary lymphoid tissue, provide essential nutrients to the host, as well as contribute to the development of many diseases such as type 2 diabetes, IBD, obesity, cancers. This is kind of the association of the microbiome community to these diseases are something we kind of want to study because it can lead to therapeutic interventions either through some sort of probiotic dietary control or through something more like a transplant, which we've seen some success in fecal transplants in C. diff patients. So classically, it's been done through statistical differential abundance analyses. A couple examples shown here. However, in the classical cases of obesity, many associations of these studies have shown to have no consensus or even contradictory results. So people have been turning to different machine learning methods with the hope that these methods can identify more complex interactions for identifying microbial biomarkers for disease. However, one thing that we noticed is that in both of the classical statistical methods as well as the machine learning methods, the only thing that was explored was abundance values, and we have a lot more information at our disposal. So in our framework, we want to incorporate kind of the most readily similarity information, which we can generate either through taxonomic information or phylogenetic information. So before we go into the in-depth analysis of the framework, we're going to do a quick overview, and there are, is there a laser pointer? Here we go. There are three kind of major components to this framework. The first is how we kind of take our abundance features and our similarity information in the form of a tree, and we integrate these into some form of input. And the model, machine learning model that we chose for this framework is called a convolutional neural network. It's been shown to have success in image processing, and we'll cover a little bit of how it works in a little bit. And then based on this model, because prediction is generally not the last step to any study, we want to break open this model and look at actually what it found. So the first thing we're going to look at is the meat of the framework, which is the convolutional neural network. And the way this works is it takes some n-dimensional matrix. So here we have a 2D matrix and a kernel that slides across the matrix. And based on the submatrix of the input it's looking at in the kernel, it takes the sum of the element-wise matrix of the kernel and the submatrix, and it generates a feature map. And these feature maps contain what are called velocities. These velocities go through some non-linear activation function. We see generally then some subsampling through max pooling, and then it's repeated again and again until you get very small feature maps, which you then vectorize into fully connected layers and make your prediction based on, in our case, disease status. However, in order to create some sort of kind of input that would make sense with this model, we wanted to treat the abundance values in a tree kind of as a canvas or an image. Because the way the CNN learns generally is by finding images or edges and creating complex shapes from edges using the kernels. So here we have a toy example where we consider this some abundance vector of a sample. And this is we use an online tool called Phylot to generate what they call a taxonomic phylogenetic tree. That's pruned at nodes if there's no OTU, but we retain the internal node just to maintain the hierarchy of the tree. And this tree is made with all OTUs present in the data set. So even if there's a 0 in this abundance vector, we'll see a 0 in this node. And the taxonomic or the abundances used will be the leaves of these trees. So we can easily just quickly assign, if this is one microbe found on this tree, we can assign the abundance here. We populate that tree by taking just a parent is just the sum of its children. And then we create some sort of image by taking the root and putting it at the top left. And then for each row, the children are added in the same order that the parents appear in the previous row. And as you can see, it kind of creates an outline of this tree. However, each subtree is now created of a unique zigzag of edges, which the CNN can now exploit to learn on. So we do this with 3 data sets. Cirrhosis, which is known to be pretty easy, type 2 diabetes, which we find an intermediate data set, and obesity, which is classically hard. And these are generated through metagenome shotgun sequencing, and the OTUs are assigned using metaflan 2, giving us relative abundance. And we compare these to the state-of-the-art machine learning methods used, which will be random forest, as well as support vector machine. We include LaSalle models, and we also include a one-dimensional CNN. So if you remember that abundance vector, this is essentially us taking out that tree representation and using a kernel over just the vector, because we want to make sure that that process of representing the tree as an input matrix gave us something. We can see that over this, we're superior to the state-of-the-art and even to our one-dimensional, which means that the representation that we generate has some sort of meaningful data that we can learn on that wasn't there before. And these were found, the increase was found to be statistically significant. So in order to understand what's going on in the model, we turn to deep learning interpretation, which is a kind of growing field of study. And we choose to look at the feature maps of the CNN model. So if you remember how this model works, the kernel slides over, and basically the abundance of this times the weight plus the abundance of this times the weight, and you do that all the way through, creates some sort of velocity where this velocity acts as a signal. The higher the signal means the more important of whatever's there was found. And so we can figure out the proportion of just this feature times this weight and the contribution of this velocity, and we can standardize it using the absolute convolution. And that kind of creates a limit between, it takes the limit from infinity to negative infinity to 1 and negative 1. So it's more comparable between reference windows as this kernel slides and to different feature maps. We do that for every sample in a certain class and take the average over that class. And then since a feature can be found in different reference windows in different kernels, and we only care about it being found once as a high signal, because that's what we expect, we just take the maximum value. And so now we have important scores for every feature based on each class. So we have some sort of perspective, and we can annotate, it's kind of hard to see. We can go back and, this tree was used to create the cirrhosis data set, and we can go back and annotate it. And what we find is that our method finds the genre OTU that was found in the original study, but we found some peculiarities. And this might be hard to see, but at this point, Bifidobacterium has a very low score of 0.02, but the parent node has the second highest score in the tree. And we didn't know if that was a false positive or if the not finding the Bifidobacterium was a false negative. And so we did some literature search and actually found that a metaproteomic study showed that this family produced glutamate dehydrogenase, which was enriched in cirrhosis patients. So even though it wasn't found in the original study, our model using the same data was able to kind of validate a metaproteomic study. And we believe that the way these scores change over the tree become an emergent property, which can be used to generate hypotheses downstream or further studies. So in conclusion, I'd just like to wrap up by saying our PopPhy-CNN framework creates a taxonomic phylogenetic tree, represents it as a matrix representation. It exploits the CNN model using spatial information to outperform other state-of-the-art machine learning approaches, and extracts features found to be important over the whole tree, allowing feature ranking and visualization. Some of the future directions would be to explore deeper convolutional layers, as well as adopting the model to handle functional network data. I would like to acknowledge my lab, the lab of Yang Dai, my lab mates, as well as NVIDIA for providing the GPU that was instrumental in this research, and the code is available on GitHub. And thank you.
Thank you, Derek. Any questions while our next speaker sets up? Can the next speaker please come up?